Does it say in the course description? It probably will say if there's a prerequisite.
All right, let's talk about cardiac cycles and pressure and volume graphs. Sweet. All right. So on this y-axis,
Really? Are you sure?
that's a horrible marker.
Oh,
All right, on this y-axis, we got pressure.
you can check. I don't think there is. I know there are.
And on this
like,
x-axis,
people like freshmen, sophomores, like anybody can take
volume.
class.
Let me see. Might depend on which one still has spots available.
in milliliters.
When so it looks like there's only 10 spots available in my class.
Yeah.
Okay, so
If you wanted to look at the
Volume of the
spectroscopy
heart.
class,
Here we go.
I know one person that's taking it. You're asking how it is.
I just don't really want to do either.
Yeah.
I might do the 290R class because that
notice
makes
right
sense
here
for
when I drew that
290R
line
class makes
that the pressure hardly changed at
Oh,
all.
that's right. I thought it was just
The
one.
volume did change.
Yeah, I've talked to Dr. B about the 290.
This is called rapid inflow.
He said that
And
it's
80%
like
of the blood coming into the heart
yeah, he said
is
it's
from
it's not-
rapid inflow, or just blood just coming in because there's a pressure.
class, you learn stuff.
It's low pressure. You know, things want to flow from high pressure
It's
to low
not-
pressure.
It's not like trying to squeeze it.
Another 20% comes from when the atria contract, right? So
I'm taking 6
you got your heart here,
so
right?
Well, maybe. Yeah.
Atria will
Yeah.
contract at the same time, and they'll fill up the
Yeah.
ventricles with heart, but with blood that
I
they
might
need.
just
So
Modern Python might be valuable.
this will stretch the heart a little bit,
Well, what are you gonna do?
and then we'll get
What's your goal?
isovolumic contraction.
Wait, if you're doing bio
Pressure will go up.
Biochem stuff, very valuable. But
Volume will go down, pressure will stay up,
maybe from that, I'm not sure.
and then we'll drop.
I'm just thinking, so I'm looking into.
This is the pressure volume cycle of the heart on average.
Internship.
Right here, we have something called preload,
With pharmacy.
or how
Pharmacy school,
like much the heart gets stretched before it contracts.
Probably, like, it'll make you look better.
What would be more valuable,
You know?
spectroscopy or Python?
With pharmacy school. I don't know, that's a good question. I don't know.
This whole line up here is called afterload, but we'll mostly pay attention to
Both
that.
would probably.
Both would probably be good.
So,
I don't think
afterload is
as useful
the
for pharmacy
pressure
as other, like,
on the aorta
research
when
things.
the ventricula contract.
There's probably,
So when the left,
I don't know.
yeah, when the
There's
left
students that
ventricle
have busy-
contracts, it's the pressure on
when
the
they talk-
aorta
When they talk about the research, they use
from the contraction.
for almost everyone. Yeah.
So let's group this up.
Python is a big research.
This right here, that's systole.
Yeah, there's
And this is
some science
diastole.
companies that like they keep their lab books in Python.
Right
it's
here,
right there. And so
You could
I
draw a
don't know,
nice
it's
little
like
line.
in the research world, there's a bigger push to like I don't know.
This is called the
not like
ionotropic
first, but it's like being cultured, like a little bit of programming.
line. The steeper this line is,
So it might be-
the harder
Yeah,
contractions
it's probably
are.
different for medical.
For
school.
example,
I don't know.
if we have,
I don't know. Yeah.
well, I guess this is also, you could call this the stroke volume. And then this is the end diastolic volume. This is the end.
I also
systolic
have
volume.
class in Brother B.
So if we
Systolic.
make our end systolic volume less, then what will happen is that will increase our inotropic line, and now our heart has to pump harder to get more blood pumped through circulation. Okay, let's draw a different graph now. Let's draw something like this.
So the kitchen is.
So this is A, this is B. We can see that in B, our preload goes up because we have more volume. And since it also increases in height, so does our afterload, so that there's more pressure on the aorta. But since they have the same stroke volume about, what stays the same? What goes down? What changes? Ionotropy. heart doesn't have to pump as hard. So that more or less is how a pressure volume chart works.
This class is taking my life.
Yeah. How to? I've heard it's pretty.
Okay, so let's talk about
Yeah.
the baroreceptor reflex. Okay, so we know that mean arterial pressure equals cardiac output times stroke volume. Okay, what does our stroke volume equal? This is N diastolic volume minus N systolic volume.
It does sound pretty
For 1 meter, you get RH.
In this case, yes.
Okay, well that's.
bad. I don't know.
So
In iron,
it's going to.
Okay,
I've only seen different.
so here we have our heart.
There's some things that are just like, wow, this isn't actually that hard. It's kind of cool.
There's the aorta,
You did lots of the intricacies.
a little thicker than that, hopefully.
Anyways, you can put any system.
And then we have this carotid sinus up
It
here.
doesn't matter how many
And then we have these baroreceptors that report to the central nervous system.
Because they don't want to mix carb.
And what they do is detect changes in pressure.
Backside attack. E2, that's going to be.
And that's okay.
We're gonna have some yoinkage.
I mean, they'll be really good at integrating, right?
And we're
Isn't it pretty integral that week?
Does it?
Yeah.
They're stretch receptors.
I mean, the first part of the class is. Now we're doing some stuff that's
Detect
interval heavy anymore.
changes
No,
in
technically,
blood pressure.
that's also.
Still just talking about.
So what will happen, let's say that we gain like 50 pounds and the length of our whole circulatory system goes up, and so our blood pressure goes up. Well, what's going to happen? These things are going to stretch. They're going to stretch, and upon sensing that stretch, you're like, whoa, we're stretching way too much. They'll send a signal or multiple action potentials to the CNS, and the CNS will be like, hey, yo, chill. And so through parasympathetic stimulation, our heart rate will go down,
You're very capable, Laura.
I'm smart.
and our peripheral resistance will go down, as our arteries will vasodilate or get bigger, right? Remember that peripheral resistance equals, I think it's like 8 VL
Okay.
over 4, no, not 4, pi R.
Dealing with an alcohol.
So this is R to the 4th. This
And we're working with a large acid and sulfuric acid.
This is what our body will manipulate to manipulate peripheral resistance or blood pressure. So,
Eat. As it's
vasodilation. All right. Say that the baroreceptors sense
Triangle.
a downward deflection in blood pressure, like they're not being stretched out enough. They'll send out fewer action potentials to the CNS. The brain will be like, hey, this ain't good. We need to up our blood pressure. Heart rate will increase, and peripheral resistance will increase, or vasoconstriction. That is how the baroreceptor reflex works. Okay, we're rolling. Okay, let's talk about mean arterial pressure. We need to go over this. Sweet. Mean arterial pressure equals cardiac output times stroke volume. Cardiac output, what's that? It's how much blood comes out of our heart. Stroke volume, what's that? That's N diastolic volume minus N systolic volume.
Yes. So we're gonna protonate this oxygen. So we're gonna go from here to here with our arrow. Because we're gonna steal it.
Cardiac output equals,
And then we're gonna get
oh no,
in.
I got that wrong, sorry. Mean arterial pressure equals cardiac output times peripheral resistance.
Okay.
This equals heart rate
Then we have
times stroke volume. This equals N diastolic
Minus.
volume times N systolic,
Okay.
minus N systolic
So
volume.
now this is what's going
This
to
equals
eliminate.
8VL viscosity length over
No. This is just gonna fall right.
pi r to the
Right,
fourth.
so because it's even. So you can only do one thing at a time.
Let's put this all together and let's talk about individually all the different
I
variables.
can't remove this either,
So mean
because that's not-
arterial pressure equals heart rate
Oh,
times N diastolic volume minus N systolic volume
How can we make the carbon cation better?
times
Great
8
question, Abby.
viscosity of the liquid
All about that.
times
I'm glad you
length
said that because I was not going
of the
to
tube
do that, but I don't
over
know.
pi radius to the fourth. That's the full equation.
Here, I got you.
Heart rate is the same thing as like frequency, right? So
I'll get one.
I'm not gonna do it. I just want to.
it's the frequency of the heart, how much it beats. Boom, boom, boom. And diastolic volume. That's how like full the heart is before it contracts. And systolic volume is heart after contraction, how much volume of blood is in there. Viscosity is how viscous your blood is. So if you have big, like thick, viscous blood, your blood pressure is going to go up because
It's gonna blow in this one.
more pressure is needed to push out the same amount of blood. Length,
Okay.
the longer the tube is, the more resistance you'll have.
Don't even worry.
And this right here is a special variable. This is the radius of the tubes, or basically a degree of vasoconstriction or dilation. And this is the one that our body will manipulate the most to maintain homeostasis. And so, in a nutshell, those are all the variables involved in determining our mean arterial pressure.
8, 5, 8, 5.
And it is important that it maintains it within homeostasis, because if our mean arterial pressure drops below 65, then it's not very good, because enough pressure, because the pressure inside our skull is like 50 millimeters of mercury, and so like it's hard to get blood up there, and so we pass out. That's why we want to maintain homeostasis, so we don't pass out.
It's still very angry.
Okay,
It's going to have a shift.
let's talk about capillaries. Sweet. Okay, so I'm going to draw a little capillary here.
Yeah, we can make two. I can make two sheer carbon cation out of this.
But if you just, if it was.
This is our capillary.
Two metal bricks.
Capillaries are so
Then a hydrogen shift.
thin, or they're just wide enough to allow single red blood cells through.
Or if that's it's on the
So
outside,
they're
yes.
very small. We'll draw the artery,
Yeah, totally.
the
Yeah.
venous ends here.
Okay.
So, what's in the capillary?
Lauren, can I stop you for a second and get your eye number?
We got little proteins here.
How you would do it in that case?
Yeah, we got some cells in here.
You're killing it, Jenna. I see why you get paid for your job. You're
We got epithelial cells in here as well that are very thin. They're kind of pancaked up in that junk. And then here, right here, this is called interstitial fluid.
20 steps.
Your red blood cells will pass out their oxygen and stuff. I'm gonna draw another little thing here. It's like a little vacuum. It's like, this is called a lymph
I'm
duct.
fine with it. Yes, goodness, they chose this. Not me.
I will say one thing.
Who are you taking
On the
this
real
class
test, you'll want to draw the Lewis structure of sulfuric acid and that.
Lymph duct.
The HSF4.
Boom. We got another mast cell here. That's about it. Okay, so our cells, they need lots of things. They need nutrients, proteins,
Brother Sarge, when I was a kid
Oxygen.
too, he was like, you know,
All kinds of
I hate
things.
this, but once this class is over,
And so,
I'll give you a list of chemistry
through these
basics. Why don't you just go
capillaries,
find some?
these cell wall here, or the cell membrane around the cell membranes of these epithelial cells, are very permeable to like all
He's like,
solids.
they'd be fine.
Like the things that can't really get through are these big fat proteins, and this is called albumin. Albumin makes up about 60 to 80% of the of the proteins in the blood plasma. There's other proteins called like, we got like fibrinogen, clotting factors,
Yeah. I just learned that that's just the relationship.
not steroid hormones, hormones, protein hormones,
That's interesting because
enzymes,
Sometimes.
the whole shebang, shabam.
Just makes sense.
So, what will happen
because every doctor
is,
talks like, I hate him, I hate him.
pressure will come into here. Not at mean arterial pressure though, because that's too high. It'll blow the capillaries right out. It'll come out in about 30 millimeters of mercury. And because we have, there's proteins in this. This is called hydrostatic pressure, by the way.
What that is.
It's barbaric.
Is that pico
I think
meters?
I just came from the.
So this hydrostatic pressure will push fluid here through the capillary, but also
She's doing
it'll push fluid out. Also, another thing that'll push fluid out into here is we have proteins out here.
Okay.
These proteins will form the
I love tossels.
the interstitial colloidal osmotic pressure ICOP.
How are you, Henry?
I'm doing all right.
You're done.
And then this will form the lymph pressure.
Let's see. I had
It's like a vacuum pressure. It's like about 3
I did
milligrams of
a little bit
mercury.
It's kind of like a
We have hydrostatic pressure, ICOP, lymph, and then this will form PCOP.
And then another lab.
Okay. How does this all sound? How does this work?
Got some food.
How do our capillaries exchange proteins while also taking proteins in like waste? Cells produce waste. They don't want to
I'm gonna try and see if we can take an exam tomorrow.
it's like you don't want to keep your garbage. So,
Yeah.
how do we do this? All right,
But,
I'm gonna write up here.
Yeah, I mean,
Hydrostatic
it's a good
pressure.
day. It just
It was about 30 millimeters
How about you?
mercury,
I'm also busy.
Yeah,
and it's a pressure
I
pulling
had
things
assignment
out.
due today, finish it like Brendan was due.
I
B
got that,
Then I have
Yep.
class, then I have
Pressure
lab,
pulling things in is about
then I have
28
exam.
millimeters
Okay.
mercury.
And now
Everything you
Pushing
do well.
things
I
in.
did.
Let's go. Okay.
ICOP
Okay, funny story. So
is about 8
for your
millimeters.
anatomy class, you get just like
Mass of your
Oh,
Pushing things
and so
out,
I
right?
studied
There's proteins in here,
on
proteins in here, right? Water follows
Turns out there was a
solute.
second PowerPoint slide that I was supposed to study.
Lymph
And it had
pressure.
cartoons, and I was like,
with mass.
No.
Just mass.
Lymph pressure is
I still
about
passed, but
3
I was like, dude, I
millimeters
didn't know
of
about that until
mercury.
15 minutes before I took these.
Pushing it out
Yeah,
as well.
it was.
So at the start here, hydrostatic pressure is so high, combined with these other things, that nutrients
Yeah.
Because like
Nutrients.
When I look at an exam, I see something new.
will flow into the cells.
Especially when you're cramming.
But how do we get rid of waste, right?
Absolutely, absolutely.
Well-
oh my gosh.
Pressure, hydrostatic pressure out here is only 10 millimeters
Got a good project.
of mercury, which makes sense. As something runs down a tube, it'll lose pressure. And so this will allow, because BCOP is so high, because its albumin concentration is so much, it'll allow toxins and other cell waste to flow in.
I'll just write on one sec.
All right, let's think about pathology here. What causes swelling? Like when you get a sunburn. Like why is it so taut? Turgor, all that. All right, talk about it. Now we kind of know how this all works.
Oh my goodness.
Yes.
Okay, say you're outside. This is like your skin, you know, whatever, skin. Sun, right here. UV radiation. It starts killing. Death. 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 Starts killing all these cells off. Now, when cells die, what happens? They spill their guts, or proteins, organelles, everything starts spilling out into this interstitial space where the interstitial fluid arrives. Now, when all these cell guts spill out there, what happens? What happens to, say, BCOP? Or ICOP, sorry, ICOP. Well,
SN mechanism.
ICOP goes way up. This will cause more fluid to flow in the capillary walls.
How far are those in this part?
This will then cause swelling, pain, inflammation, all the symptoms that are associated with a pretty severe sunburn. And this is one example of edema. And there's thousands of different ways in which this homeostasis of the capillary action might get messed up, causing edema of some sort. And so, as a physician, it is important to understand exactly what's going on here so that we can diagnose and we can give out necessary treatment.
And then